Alright, now let's divert our attention into the mitochondria, where we will find the electron transport chain. Now regarding this process, there are quite a few fancy figures out there, including this one from the book, but I'll break this down and make it as simple as possible as we walk through this process. Now we start out here with our 5 major complexes, named Complex 1, Complex 2, Complex 3, Complex 4, and Complex 5. And each of these have a unique function. And just to orient ourselves, here we have the inner mitochondrial membrane, with the mitochondrial matrix located here above the membrane, and our inner membrane space located below the membrane. Now our pre-made activated carriers, like NADH as well as FADH2, will dump their electrons at these complexes to ultimately create ATP. And where exactly did we make these carriers? Well, remember that we had some pre-made NADH in glycolysis using the enzyme pyruvate dehydrogenase, and also we made some NADH at various steps within the TCA cycle, and also FADH2 was made in the TCA cycle. Now NADH enters at complex 1, and FADH2 enters at complex 2, which happens to be the enzyme succinate dehydrogenase. And can you think of where else you've seen this enzyme? There you go, that's in the TCA cycle. Now as electrons pass through these complexes, they create a proton gradient that is used to drive the production of ATP. Now complex 4 is also known as cytochrome C oxidase. And this complex is involved in the transfer of electrons to the final electron acceptor. And do you remember what the final electron acceptor is for the electron transport chain? There you go, it's oxygen, which actually turns into water after it's accepted its electron. And without oxygen, there is no final electron acceptor, so the electron transport chain cannot function. And this means that the body would have to rely on anaerobic metabolism for ATP production. Now complex 5 is also known as ATP synthase. And ATP synthase here uses the hydrogen gradient that complexes 1, 3, and 4 made to produce 2.5 ATP for every NADH and 1.5 ATP for every FADH2. Now since FADH2 starts at a later complex in the pathway, it isn't able to pump as many electrons across the membrane, so it only makes 1.5 ATP. Now complex 5 is going to take ADP as well as a phosphate and form a high energy ATP molecule. Now that we know how this process works, we can step aside and introduce 3 scenarios where oxidative phosphorylation becomes poisoned or inhibited. The electron transport chain inhibitors, such as rotarone, cyanide, Annamycin A, carbon monoxide, and azide directly inhibit electron movement from complex to complex. This prevents the formation of a proton gradient and therefore synthesis of ATP. Now rotarone inhibits complex 1, and Annamycin A inhibits complex 3, and carbon monoxide, cyanide, and azide all inhibit complex 4. Now next up, we have ogliomycin, which is an ATP synthase inhibitor. Therefore, it inhibits complex 5. Now ogliomycin here will allow for a proton gradient to build up, but there won't be any creation of ATP. And if you cause a logjam in the electron transport chain, you're going to get a buildup of NADH as well as FADH2. And last of all, we're going to talk about the uncoupling agents. And how do you think these work? Well, the uncoupling agents, such as 2,4-dinitrophenol, which was once used as a weight loss agent, as well as aspirin and the protein thermogen, which is produced in brown fat, all increase the permeability of the inner mitochondrial membrane. And this will result in loss of our proton gradient, as they will flow back through our inner mitochondrial membrane. And since there's no proton gradient, you can't generate any ATP. Now this results in increased oxygen consumption because the electron transport chain will attempt to compensate for the increased permeability by revving up its activity. Still, with this, ATP cannot be created without the proton gradient. And this increased electron transport activity is instead dissipated as heat. This is one reason why aspirin overdose can cause fever, as well as why babies and hibernating bears can stay warm, since they have lots of brown fat. Now if we were to put all this together in one image, we have our figure from the book, and this is a great place to review this process. Well, alright, now let's take a shot at a flash quiz. A 4-year-old girl swallows some cyanide-containing poison from under the sink. How does this affect her ATP production? Well, remember that cyanide directly inhibits the electron transport chain, specifically at complex 4. This results in a decrease in the proton gradient and blocks ATP synthesis. Therefore, the body isn't able to create ATP via oxidative phosphorylation. Well, alright, that'll wrap up the electron transport chain as well as oxidative phosphorylation. See you in the next video.